Sorry for this.
Use the microphone.
Yes.
Oh, sorry.
The light's really good.
Oh.
Oh, and it's, if it's in the volume. Oh, I can control this.
What can propagate this?
Okay, so you're going to do that?
Yeah, wait a
Can you hear me? Okay. So I'm going to talk about sort of pattern formation issues in a growing bacterial colony or colonies. And this work, I'm from University of California, San Diego. I work at the Institute for Nonlinear Science. But this work was done in a close collaboration with a group of people at Bioengineering, who I am working with on some other things related to gene regulation networks. And this is a lab of Jeff Misty. And so some of the work which was done here was done in collaboration with Richie Wolfson, who was a postdoc at INLS with me, and then another theoretical postdoc, William Mather, who is currently there at Bioengineering, and two experimentalists, Scott Cookson and Octavio Mondragon, working on some parts of this research. And these are some references. So one paper was recently published in PNAS on the first part of my talk, which is related to ordering of populations and the streaming business. We are preparing the paper right now. Okay, so as I said, I'm collaborating with this experiment on some other issues related to gene regulation. So the typical experiments which we are doing there, we are interested in gene expression using fluorescent measurements in microfluidic devices. And so the typical experiment works like this. We have a channel, relatively shallow, like a 3-micron channel where the nutrients come and flow, but then and possibly cells too, but they don't stay put. But then we have an area which we call a trap, which can have a different shape. You can design any shape you want. It can be open from all sides, like here, or it can be semi-closed, which is a shallow area, which is only 1 micron deep. And in this area, bacteria, if it gets there, it gets stuck. And I should mention that these bacteria are sort of not very common in wild type, but they are typical for laboratory studies in signaling and gene regulation. These are non-motile bacteria, so they don't move by themselves. They basically are like granular rods, and which is kind of nice to me because I've been working with, for example, with Ashad Kudrori and by myself on granular rods for quite a while. So I looked at that and said, oh, that's an interesting granular system. But it looks similar to granular, but it has a big difference because cells don't stay stationary; they grow and divide. And what happens is that initially you get very few cells inside the trap, maybe 1 or 2, but then they start to grow, and eventually they fill up the trap completely. And it doesn't take too much time to do that. Cells divide at every approximately 20 minutes. So after a few hours, you have a completely filled cells, and cells start to go out of the trap. So that's why it's open, because you don't want cells to be jammed to death there, but so they have a room to escape. And once they escape, they are flown away by this nutrient flow to the wastewater. So inside of the trap, we have a quasi-stationary condition, but dynamic condition, because cells are growing at constant rate. So when we looked at this is an example of the typical movie, this is just a bright field, so we don't see any fluorescence, but just if you look at this, you can notice something interesting. You can notice a couple of interesting things. One is that there is a local orientational order. You see that a lot of cells are oriented along the same direction. It's not a global order, it's a local order, so the orientation is different parts, but it's a clearly nematic-like structure. So that's one thing. And the second thing, if you look more closely, you see that cells are escaping. So this is, I should say that this is so only one side of this trap. This is a part of the whole picture. So this is a solid wall. So cells don't escape this way, and they don't actually escape on these two sides. So only this side is open, and they only can escape in this way. And what you can see there is that what you could see is maybe I shouldn't take too much time, but what you could see is that the cells do not escape uniformly through this wall. what you could expect is that they all go out at the same rate everywhere across this channel. They actually escape in some kind of streams. So there's a streaming. There is this large-scale flow in homogeneity. So they kind of go here, they go there, but they don't go so much here. But they go much slower rate. And it seems like an instability, and we wanted to understand what kind of instability is this, what causes that kind of large-scale structure to form in the system. So there are these two issues, and I want to address them both in my talk. Okay, so just a very brief introduction, which probably already, maybe Arshad already did something like this. I'm interested in granular rods because this is an interesting variation on the theme of granular physics, which I've been interested in on. And then it's actually very important. It has lots of applications on all kinds of scales, from very large scales, like in industry, pharmaceuticals and stuff like that, down to very small scales where you have bacteria and viruses which have this shape of rods, very typical. And even you can view biomolecules, these microtubules, you can view as little but very stiff granular rods. So you can develop some theories, which we did some work on that, on how these rods are actually interacting with each other, but in a non-trivial manner because this interaction is mediated by molecular motors, and so it's not typical granular rod collisions. Anyway, so there's a lot of interest in granular rods right now, and this work on bacteria sort of occupies a certain niche in that whole activity. And speaking of ordering, I mean, there is a very well-known Ansager type equilibrium entropy-driven transition. It's well known that if you increase the temperature for very low temperatures, the rod-like molecules, they form symmetric crystals, and then you increase temperature, and then you lose positional order, but you still can retain orientational order, and you form pneumatic crystal, and then you have a transition to an isotropic state. So this is entropy-driven, and it's well understood, and so forth. So the question is, and this is an example of active granular rods. So granular rods you can supply temperature to them by driving them because you have dissipation, so you have to drive them somehow. And so typical way of driving them is shaking them, and Arshad probably mentioned some of this in his talk. So you can have very interesting order states there, which are different nature and different mechanisms from this equilibrium Ansager type transition. So you can have either rods oriented in a third dimension, so you go beyond this two-dimensional state into an ordered, like one layer symmetric, or you can see a symmetric order in a monolayer, also vibrated, or static, or some other states. Anyway, so this is non-equilibrium phase transitions, but in the typical granular rods. I'm interested in this kind of system and the mechanism of ordering in this system. And it appears to be quite different because you really, again, if you take a single particle, as I said, it doesn't move. There is no thermal noise, doesn't apply. It's big enough and it's squished enough by the wall, so it doesn't really shake. But they start to move when they're pressed against each other. So this transition, apparently, at least all the dynamics here is driven by the growth and by the pressure which they exert on each other. So it is somehow related to that kind of biomechanical interaction. And so we decided to look a little bit in more details at this, and instead of looking at these big channels where you got this two-dimensional inhomogeneities and whatnot, we looked at the same system but in a narrow channel, so hoping that we'll have a one-dimensional flow and it will be easier to understand. So we redesigned a little bit our microfluidic device, so we had these multiple flows, and then we could look at multiple channels, and we could look at each channel. So it's surrounded by solid walls on both sides, and it can escape, cells can escape in both directions. Okay, so these are some of the movies. So we can start with a single cell, and so it creates an expanding colony, or we can try to spread the cells inside of the channel and then let it grow from there, and that is a more like you start from random but sparse initial conditions, and then you let it.
How much time it takes?
How much time? I think it's about 3 hours, the whole movie here, in both cases, something like this. I'm sorry, there's no time. So we can see there, but so the order, you can see that clearly most of the cells are oriented along the channel, which is what we believe is that the core of the mechanism. Although you can also see that it's not quite a perfect order still, even though this channel is quite narrow, it's only about 25, like 30 micron across, so, and each bacterium is 1 micron approximately in cross section. So it's pretty narrow, but still you have some remaining disorder. So it was not narrow enough to consider perfect order. Still, we could do analysis of this because we could do PIV on the velocity field, and we could do actually an image segmentation, so we can determine the local orientation of order. So this is an example of how we can measure density field, which is shown in red, and velocity field, which is shown in blue. And you can see that initially there is not much in terms of the flow. There is a little bit of the flow on the outside, but eventually, once the channel becomes completely packed, you see something like a linear velocity profile, which is kind of makes sense. If you have cells are growing at the same rate in the middle, it's very easy to see that if the density becomes constant, once you reach the close packed density, you have to have a linear velocity profile to equilibrate the growth. So the divergence of V is equal to alpha. That's basically how it comes.
So that coordinate is left-right in the picture?
Yes, that's across the picture, sorry. So the density is basically constant, and that basically requires the system to have a linear velocity profile. Although the zero does not necessarily have to be in the middle because it's a different.
It's the number density?
It's the density. I think it's just the density of cells.
Number per unit area.
So the area, yes, the area occupied by cells.
It seems that the cells at the edges are bigger than the cells at the center.
Yeah, and I will talk about that more in the second part. So yeah, that's it's a very interesting, very good observation. but we measured just the density of the area. Anyway, so as I said, in addition to those kind of bulk measurements, you can actually see individual cells when you work with high resolution microscopy, and you can measure individual orientation. You can estimate the order parameter, the tensor, because there is no direction associated with cells. We have a tensorial order parameter, and then this is the magnitude of that, which is basically defined like this. And in a disorder state, Q is equal to 0. In the ordered state, in the complete ordered state, it goes to 1. And this is one of the examples of what the order parameter is doing in this channel, starting from random distributed initial conditions. So it starts from pretty low values of 0.2, 0.3, and then it goes to not to 1, but to much higher values of 0.6. Okay, then so that was our experimental observations. We also decided to augment them with molecular dynamics simulations, since we already had the code which we developed for granular studies. It was published in this work. We just basically had, I don't have time to explain in details. It's a pretty straightforward soft particle simulation algorithm. But we had to add a little bit to this to accommodate the growth. So we basically, each cell is growing at an exponential rate, and once it reaches twice the length, it divides. And we introduce some kind of randomness in this division process. So the critical, the length at which it divides is not exactly the same, but it's distributed. Otherwise, you will have this spurious. Every cell will divide at exactly the same time. But that's easy to mitigate. Anyway, and this is an example of these simulations, similar to what we have in experiment. We have solid walls on both sides, open with boundary conditions here. So as soon as cell reaches that part, that side, then we just take it away from the simulations. And the color here indicates the orientation. So it's red when it's oriented along the channel. It's blue when it's oriented perpendicular. And you can see that the channel quickly becomes almost completely red. There is very few patches of disorder, but not much. But they are, interestingly, they are persistent. And you can see that if you make simulations bigger, okay, so this is some snapshots. This is some analysis. Of course, in this case, we know everything about the system. We know the orientation of every cell. We know all the forces acting on each cell. So we can compute the pressure tensor, and it's interesting that pressure was isotropic initially. As soon as the cells are oriented along the direction, you have strong anisotropy in the pressure tensor, which is kind of to be expected because when they're growing, they are pushing only along their orientation, and so you have a longitudinal part of the stress tensor is much higher than the transversal part. And you can have order parameter as a cross section, which and the average order parameter is a function of time. Density, so you can measure everything basically, and it all corresponds, at least as concerns the order parameter and all this transition, it corresponds pretty well on a qualitative level with the experiment. So you really see that you don't need to put in the system anything beyond just pure mechanics, and there is no noise, there is no thermal fluctuation, and you see very quick orientation of the cells along the flow. And okay, I'll skip that. It's just basically the same thing. done with randomly distributed initial conditions. What's interesting is that if you have a big system longer than what I showed you later, you don't have, even if you wait for a very long time, you don't have complete order. You still have patches of disorder, even though the order parameter is not zero, it's high, and you have orientation on the channel, so it's predominantly red. But you have these patches which are persistent, and that's one of the questions which we are actually puzzled about. Well, maybe not puzzled, but we are interested in doing it. But it's still, I mean, now I think only now we are starting to get some idea of what kind of instability is that, and that's so this is still work in progress. I'm not going to talk about this, but it's an interesting buckling type instability which occurs there. So this is an example of what, if you have a short system, you have complete order, like shown here. If you have a system which is with aspect ratio 4, you have these patches and they don't go away, so they regenerate completely. And we believe that that's related to the fact that pressure in the middle is high enough, so that because of there is this resistance to escape. So pressure, to accommodate this linear velocity profile, the pressure inside has to be parabolic, as you can imagine, right? And so pressure in the bulk becomes very high, and that's where it starts to buckle. Okay, so you do some analysis, but that's, I don't want to stop here. So you try to explain it theoretically, and the theoretical description can be based, for example, I mean, you can develop different levels of complexity in the modeling, but the level we chose to do that was using these pneumodynamics equations, which were put together, proposed by Toy Edwards, and there are some other people, Goldberg. Right, it was an equation similar to that. So it's an equation with a tensor order parameter that basically fluid dynamics coupled to the dynamics of this object Q alpha beta, and there are several terms here in the right-hand side. Some of the terms are related to basically just shear orienting the order parameter. So you turn the order parameter by the shear, and those are these terms. And then you also have the term which is related to the elongation of flow, which basically produces order if there is no, even if the order parameter was 0 initially, it will be starting start growing linearly because of that term. And then there is this normal term which is proportional to the molecular field, and that is the term which usually drives the standard Untitled type transition. But the rate of this term is usually proportional to the temperature. So you need some temperature for the entropy to minimize the temperature, the entropy. And in this case, as I said before, entropy doesn't seem to play any role because the temperature is 0. So the system does not explore all the configurations to minimize the entropy. So basically, what we did is that we started from this model, but then we added something and we deleted something from that. So what we did is okay, this is a continuation of this. Of course, we have to couple this with the momentum equation, and we have to write down the standard expression for the stress tensor. But then we had to add the growth, and the growth is in this density equation. We have this additional term alpha rho, which leads to exponential growth of density. And then, of course, once density reaches comes close to some critical density, then the pressure starts to grow. So we have a constitutive relationship between the pressure and density. And so once pressure starts to grow, then there's a pressure gradient, and that pushes cells away, and that compensates this growth. So there is this term here. When the flow appears, the gradient of the flow compensates that growth. So there is this part, and also we deleted all the thermal motion in that. So we deleted this gamma H term, and basically the stress-strain relationship become much simpler. So we have this, and this critical density actually is a function of order parameters. So if you have a disordered set of rods, the critical density, closed pack density, will be less than when you have a perfectly ordered set of rods. So we also took that into account. So that's the model which you can simulate, and you can either do it just in this form, or you can simplify it and go to one-dimensional case because we have this narrow channel, so everything depends only on one coordinate x along the channel. Or we can do it even simpler. Actually, there is an ansatz you can use. If you assume that the velocity profile is linear, which it is, because if the cells are basically incompressible, then you can plug the ansatz. The pressure is actually parabolic, has to be. And then there is a set of ordinary differential equations for the density when it reaches the critical state, for the only one component of the order parameter, this x component, and for the amplitude of the pressure and for the amplitude of the shear. So you have this system, and then you can integrate this, and you can see that actually the dynamics you can fit the parameters to, there are not so many parameters here, you can see. But you can fit basically all sets of data from the numerical experiments, not from the original biological experiments, but from at least from our numerics, you can fit all the data pretty well, actually. You can see even the subtle features like this overshoot in density, in the shear and density, you can see that. So you can basically explain all the features of how the order parameter grows. I'm seeing what is so this is my order parameter, and there are two different types of models. I didn't mention that, but there are we had different, because that's what we see in experiments. In experiment, it looks like in the bulk the cells are growing slower than they grow in the periphery, and we believe that one of the explanations, there may be several explanations for that. And one is that when the pressure becomes too high, they just don't have, they slow down the growth. And of course, there may be another explanation because of in the bulk, the nutrient can be also low, and so that also slows down the growth. So, but it really,
you can have aging too.
Yeah, but it does for bacteria. It doesn't. I don't know. I don't see that that's the case. I mean, for things like more higher organisms like yeast, that's an explanation.
Asymmetrically divide.
We didn't see evidence of that, but maybe this is a very model bacterium. This is this laboratory strain, KP1 or something.
Sorry, which are the free parameters?
Which are the parameters? Well, there is a parameter with this B, which is orientation. I mean, some of them you can normalize. Like this is alpha is the growth rate, so that's a scaling that scales your time, so you can take that away. So there is this parameter D, which is the rate with which basically the order parameter is growing because of the shear. That's I mean, you can calculate it, but we treat it as a free parameter. And then there is this relationship, the constitutive relationship, which relates pressure, how it grows with density. So that's A. That's another parameter. And then there is friction coefficients here. So mu is the friction. So your pressure has to be balanced by friction. So that's basically all there is there. Three parameters. Okay, and we can fit the density growth. So it initially grows exponentially, then it saturates at a certain level, and this is the shear. So it goes to a stationary state, which goes to 1 here, and then it goes. So blue is pressure independent growth. So if the growth is pressure independent, it has to saturate at 1 because of the mass conservation. If you have a pressure dependent growth, it saturates at a lower value because in the bulk cells don't grow very fast. Actually, they don't they grow very little, and that actually affects quite a bit other things like the pressure tensor. Pressure tensor becomes actually isotropic in the case of symmetric growth, of this pressure dependent growth.
So did you put that in explicitly by letting alpha depend on P?
Yes.
Yeah.
Yeah, I didn't put it here, but okay. Anyway, so let me stop here because how much time do I have?
8 minutes.
Okay. Okay. So now back to that first movie I showed. So if you look at it again, now I want to focus your attention on these streams which I curved here. So you see that cells are flowing kind of here, so flowing but not as much here. And maybe you can see that that's your imagination, but if you actually do some analysis of this picture and then you do particle image velocimetry, you really see streams. So this is a snapshot from that movie, and you just take 2 consecutive snapshots and then do PID, and then you see this quite well-defined streams.
Quick question. Is it also true in this one that the particles change size, or is that just an imaging?
No,
they
do
look
change
really
size.
small.
Oh yeah,
at the
they
top.
do change size, and that's the thing I wanted to connect to what we're seeing here. So this is the same PID, but now we have this is a time coordinate. So at every time we can do a PID, and that shows you basically color indicates the velocity in this lower, let's say 1/10 of the image. So you integrate transversal velocity of the cells across a certain slab here, and that's what you see. So you see originally there is a big patch here, but then kind of disappeared. But then there is another stream here. But you see there is a lot of intermittency here. So it's not a uniform stream, but that's really what happens here is that there is like a stick slip kind of motion. But it's a very non-uniform across. So you see, and some new stream appear here, then they merge, and so there is this kind of birth and merging of streams. And this intermittency in this direction, we believe is related to this stick slip kind of motion. So there's a if the patch of the cells has go away as a whole, and there is some room for them to grow before there is a new stream of accumulating.
So is it a steady state? So are
This
there
is
some characteristics?
Yeah, you can wait for, it's a quasi steady state. So you can run this experiment for a very long time, and that's more or less what keeps going on. That's what your question was?
Yeah, but I see that the velocity profile changes, but probably there is something that remains constant with time.
Well, the overall escape rate, if you integrate this, it more or less remains constant. It may fluctuate, of course, but it remains constant. So the overall number of particles which are leaving the cell, that's determined by the average growth rate inside of the chamber, and that remains constant, but they don't escape everywhere uniformly. So they escape here more and there more, and that sometimes they escape in the middle. So that's the streaming which I'm trying to explain. And then, indeed, what you notice here is that the cells in this part are much smaller than here, and that's actually is very important for the mechanism which I'm trying to put forth. So this is an example of the trap, which is not much, but somewhat deeper than the other one. The previous one was 1 micron deep, and this one 1.6 micron deep. And in this case, you can see that it's much more uniform. So this is the same measurements done here, and you see very little in terms of streaming here. So with the same scale of the colors, you see that it's more or less uniform. So the question is, what is different? Why narrow channel produces streams, and this wide trap does not produce much of the same? Even though you still see the same effect that cells in the back are small and cells in the front are big. So in that case, it's the same effect, but not much in terms of streaming. So this is another type of experiment with a very large trap, narrow 1 micron, but it's about 250 microns across, much bigger than the other one, and it's very long. It's only a small piece of that. The flow is, I think, in this direction surrounding this trap, and you can see this very well-defined stream. This is a bright field superimposed with fluorescent imaging, so you can see these large streams better here, and you can see that all across. So that seems to be a very common phenomenon, at least in these laboratory experiments which we observe, and we believe that it may have some relevance to cells in biofields when they accumulate in these tight spaces and they escape from them. So, yeah.
What's the coloring?
Well, this is some constitutive protein this is GFP was put under control of some constitutive promoter, and that was expressed in principle in every cell at a concentrate, but it turns out that it was more expressed in the middle, because of the certain level of nutrients and whatnot, and the cells which were expressing GFP were carried away by the flows. Okay, so what's the mechanism of the streaming? And then, so what we see in those movies, as you already noticed, that the cells in the back are small, and the cells in the close to the opening are big. And why is that? It's more or less clear, because cells in the back, they have much less nutrient than the cells are in the front, because nutrient has to diffuse through that colony, and it gets consumed by the colony. So it actually depletes in the back. By the same token, the amount of waste in the back also accumulates, because it has to be diffused away, and the diffusion is impeded by the cells themselves. So presumably there is a strong gradient in the growth rate, in the growth conditions. So they're much better in the front than in the back. Okay, so let's look at this side view. So this is the back of the trap, and this is the front. So here is green. Supposed to be green. This is where the conditions are good, and this is where conditions are not so good. So if there is a cell here, then it will grow to a certain size, and eventually its size will saturate at a certain level. And if it will stay stationary here, and it will grow, it will grow to a larger size and stabilize at that level. But they don't actually sit still; they're actually moving because they're pushed by other cells. So if the cell gets to a small cell, new small cell appears here, it will be gradually pushed away. And so what happens is that if it moves quickly, it will grow, but it will go away before it actually grows so large, so it will not get stuck. But if it moves slowly, like in this example, it will actually grow to such a big size that it will get stuck in some place. And so, And so what happens is that if you have this uniform flow, but now you can impose small perturbation to that flow. So if you, in some place the flow is slightly slower, than outside, then cells there will grow bigger and then will get stuck, and that will slow down the flow even more. And if it will slow down in that particular location the flow even more, then cells which are in the back, they have to bypass that jam, right? And they go to in the more narrow opening, so they will go faster there because the space, the cross section for them, effective cross section becomes smaller, and so they will grow even faster, and for them it will be even easier to escape. So there is a mechanism of this instability. So basically, if you have, like, you can imagine you have two doors here. If one door gets jammed, then everybody will go through that door, and they will go quickly, so they don't have a chance to grow big, and so they will have easy time to escape through that door. So that's what happens, and then we can test this hypothesis in molecular dynamic simulations, and it's the same model as before. We just added one more equation here. So okay, this is just using equations. This is dynamics of the cell length. So cell length, so they grow and divide, but there's also another cross diameter of the cell, which now is a function. So it grows with a certain rate up until certain equilibrium size, which depends on the distance. So the C of R is the equilibrium size of the rods if they were stationary at a certain position. And so C of R, C is small in the back and C is large at the front. But because gamma is finite, so if they don't grow very, very quickly, then again this delay in the growth because of the motion will actually lead to the fact that they will not grow to the stationary C of R. They will escape at the smaller size. Okay. So you can simulate this, or you can simulate continuum equations and or do some analysis. And that's what we did. So basically, that's the continuum version of those equations. You have fluid dynamics, and here we, for time being, we forget about the orientation of the cells. Assume that the cells are spherical. Normal fluid dynamics, and there is this, again, divergence of V equals to alpha. We assume that there is incompressibility, and there is another equation for F, this cell diameter or cell whatever, as a function of distance. But then there is a friction term, and so the friction term is a nonlinear function of this F. So the friction coefficient depends on the size. So if size becomes large, friction becomes large. And it can be actually very nonlinear because if cells are not touching the walls, they can kind of escape. Once they are touching the walls, all of a sudden friction becomes very large. So that can, in principle, be a very strong function of that. Okay. So, all right. So now we can either do simulations or we can do both analytical studies using those continuum equations, and they actually match very well. So this is an example of our discrete simulations. And this is, depending on parameters, you don't have to have an instability. So in this particular choice of parameters, there is no instability. There is just uniform escape, and color here indicates this F, basically the size of the particles. And you can calculate the stationary state very easily from those equations. And then you can study various configurations, both analytically and numerically. For example, we started from analysis of narrow flow again. So when there is one-dimensional hope for one-dimensional solutions, and that's what we did. So if there is a small cross-section, then you start from growing, and so this is this analog of the previous slide. So it's a one-dimensional solution, and this is the velocity, absolute value of the velocity. It's a linear profile, but symmetric. And then this is F. So F is minimal in the middle, and it grows to the edges, as you can expect. And velocity in the middle quickly approaches zero, so that means that it's a symmetric solution. Now, if you increase the parameter B here, it was 10 in the previous slide, so B is basically the gradient of the concentration of the nutrient there. So it becomes, this is from continuum equations, this is molecular dynamic simulations. You see that solutions are symmetric. So so it's jammed here, and so the velocity is basically oriented towards the other door. And then you can also see that here, so velocity is actually shifted, profile shifted from symmetric state, and the density is also non-symmetric. And there is some kind of overshoot. And you can even have in the longer channels and very strong nonlinearity, you can have oscillatory solutions, where it is sort of goes left, and then goes back in the middle, goes to the left door, goes to symmetric state, and so forth. So you can predict that in analytical theory as well. So this is symmetric, then again grows. It gets jammed here, symmetric, and so forth, and it just goes further. that's how it looks in continuum theory. Okay, and we did the complete bifurcation. Yeah, I'm well finished. So we did the bifurcation analysis of this. It's actually a pretty interesting bifurcation diagram. There are several types of non-trivial transitions within symmetric state, asymmetric, oscillatory, with some interesting bifurcations, but I don't have time to talk about that now. And then we went on to look at the spatially extended system. We can do linear stability analysis, and we can show that we calculated instability growth rates, and we could demonstrate that there is a lateral instability in the system, which actually leads to streaming. And this is an example of the system, again, discrete simulations here. And this is a space-time diagram similar to what I showed you in the experiment, but this is from numerical experiment. So I'm slow because it's a very long movie. Just because a while it's like a week worth of calculation, maybe not really. But eventually, you'll see you start from a single cell, so it grows. And it's exactly that simulation which was used to generate this pattern. You kind of can see that this stream is, I think, is this part, and that stream is this one, and then you start to see another stream developing over here, and this stream start to travel. So we don't have to wait until this movie goes. You can kind of see what is going on here. So this stream can merge, and new stream can appear. So it's an interesting spatial temporal dynamics, which seems to be very similar, at least on this semi-qualitative level, to what we see in our experimental setup. And okay, let me skip that. So it's interesting that this aspect ratio, even though I ignored the orientation, aspect ratio plays a strong role in here, because if you have longer cells, then this orientation it amplifies the streaming instability dramatically. So you see that it's much more dynamic and much more pronounced than it was for short rows. This is aspect ratio 5. Okay, and let me skip this. You can see symmetry breaking instabilities, interesting shape. This is when the channel is open from both sides, so you can see structures like this. Okay, skip that, and directly to conclusions. So my conclusions are that so this very simple system, it's almost like a granular system, but with a slight modification of the cells growing and dividing, leads to quite interesting spatial temporal dynamics. It's basically an active granular matter, even though most people say the granular matter is already active because we have supply energy. But this is sort of intrinsically active because they're growing by themselves. And what we saw is that there is this expansion flows which are inherent in growing populations because they have to escape. And once they start to escape, they orient themselves along the flow, and that leads to a local orientational order in populations. And then we see that if you have different growth conditions in different parts of the population, that actually can lead to interesting pattern-forming instabilities like this streaming which I described here. And so we did both experiments and molecular dynamics and analytical theories, and they seem to match. Molecular dynamics and continuum theory match pretty well quantitatively, but of course, with actual experiment, we have not so much quantitative, but qualitative. And there are certain directions which we want to move in because we want to add some physical effects which are relevant for real bacteria in the world, not just in our lab, and that usually in biofilms. There's adhesion, there is all kinds of other complications, chemotaxis, and that would be interesting to see how these effects play a role in the actual biofilm. But that's good. Thank you very much.
Thank you very much. Questions? I'm told that the microphone is somehow distance sensitive, and you're supposed to hold it close to your mouth. Is it on?
Yes.
Okay, so you hold it close. If you hold it far, they can't hear you. Who has a question? Other questions? Yeah.
I have a quick question. How do you get the bacteria to not swim as they grow?
Well, this is a special strain. That just doesn't have flagella. It doesn't have the apparatus for making flagella. Yeah.
Is the boundary sharing to the side? So when it grows, the bacteria...
You mean when you have a channel? Yeah, there is some of that. In the molecular dynamic simulations, we just made it so there is no sharing, just to simplify life. So the friction between bacteria and with the walls was very small. The only friction was with the bottom and top of the chamber. In the real system, there is some kind of shear, of course, across the channel. Yeah, something like that.
Why don't you get quantitative agreement with the experiment? Do you understand that?
Or quantitative?
What is missing? Can't you just fit the parameters to the experiment?
We didn't attempt to do that. We probably could, but it
In the
looks...
simulation, you know everything. In the experiments, and the parameters you don't know.
Could you repeat the question?
Why don't you?
He's asking why you don't get quantitative agreement with experiment.
Well, experiment is kind of dirty in the sense that not all bacteria are pretty different from each other. So some of them, for example, get sick and they just grow very long, like long sausages, and fail to divide. That completely throws away the whole thing. So things like that happen. And also there is this stick-slip kind of phenomenon. So they kind of grow and then they escape. So there is some nonlinearity friction which we didn't take into account. And also there is this extracellular matrix which probably is developing there. So it's not, it's presumably non-Newtonian fluid of some kind. So we could, yeah, but we cannot carry it.
Both in hydrodynamics and in these simulations, you have some scale, so some K,
The wave number?
of these instabilities. They have the same, we observe the same,
I'm talking dirty.
the same length scale in the
Typical. I mean, if you have a small chamber, then the scale is actually determined by the depth of the chamber. In the deep channels, like what I showed here, there is an effective depth because also the other complication is that the growth rate again is depending on the distance. So effectively, the cells in the very back, they don't even grow very much, and they grow very little. So you have this effective depth, and so the size is it's like a fluid. You know, if you have there's a because of the incompressibility, the transversal scale has to match the longitudinal scale. So that's what. But that scale is determined by the growth conditions.
Other questions? If not, let's thank the speaker again.